liftoff of the Space Shuttle Discovery with the Hubble Space Telescope, our window on the universe.
In 1990, a space shuttle was launched carrying a special payload.
Roll program.
The Hubble telescope.
Roger, roll, Discovery.
Once this remarkable piece of equipment was working properly, it was able to look into the farthest reaches of the universe, picking up the faintest glimmer of light from deeper space. Now scientists are using the same technology on us to look into the farthest reaches of the human cell. Rural Connecticut has been the scene of a recent revolution in genetics. Biotech pioneer Jonathan Rothberg has developed a new system for sequencing DNA. It has sped up the process 100-fold.
DNA is the code of life, and it literally is 3 billion letters from your mom and 3 billion letters from your dad, and these letters are in long strings, just like words or paragraphs or chapters of a book, arranged on your chromosomes. It is your computer program, and this program has evolved and been passed on. I find nothing more fascinating than understanding and working out and decoding the blueprint for humans.
Rothberg's breakthrough in sequencing DNA was to code it with the firefly enzyme luciferase. When loaded into an FLX sequencing machine, the luciferase reacts with a series of 4 different chemicals. Every time there's a reaction, there's a tiny flash of light. Each flash relates to a letter of genetic code, and a camera records them all, gradually building up a composite picture of the entire genome.
We need a camera that has very high resolution, that has many pixels, but also is very fast and is very sensitive because it's very tiny amount of lights that we output. But by using an astronomical camera, we can actually capture these many hundreds of thousands faint glimpses of light.
The cameras they're using are the same as the ones developed for the Hubble project, designed to pick up the dimmest light from the most distant galaxy. A new company, 454 Life Sciences, is created to develop the sequencing technology. It announces itself to the world by delivering the first personalized genome.
Jim Watson, the first personal genome.
They choose James Watson for the honor, one of the original discoverers of the structure of DNA. It takes them just 2 months to unravel his entire genetic code. But sequencing the Neanderthal genome promises to be much tougher. Despite the technical advances, the challenge is enormous. Even the star sample, Croatian bone V80, has DNA that's at best only 5% Neanderthal. The rest is DNA from microbes, insects, or even modern humans. To get at the Neanderthal genes, they have to sift through all the other genes as well.
The challenge of deciphering the Neanderthal genome is very akin to taking these shredded novels, which have War and Peace, mixed with 20 other novels. It's further complicated by the fact that I don't know the contents of those other novels, and I don't have a reference to this original Tolstoy. So from scratch, I have to pull out all of these fragments, read all of these fragments, and sort out which ones belong to War and Peace and which ones belong to these other novels.
This is precisely what's involved in sequencing the Neanderthal genome. Looking for 3 billion letters within an enormous pile of junk.
We don't have the luxury of reaching in and pulling out just the Neanderthal words, paragraphs, or letters. We have to sort through 60 billion of these letters until we have 3 billion that belong to the Neanderthal. We then have to assemble that into Neanderthal genes, Neanderthal chromosomes, and ultimately the Neanderthal genome.
In October 2007, the Neanderthal Genome Project announces its first significant result. Researchers find evidence of the MC1R gene in Neanderthal DNA.
MC1R is a gene that influences pigmentation. In particular, it can influence your hair color. There's a mutation to MC1R in living Europeans that gives people red hair, for instance.
In modern humans, the MC1R gene sits on chromosome 16. And in 80% of people with red hair, this gene is faulty. Now it seems the same was true of Neanderthals, suggesting that they also may have had red hair.
This gene is the first piece of evidence that we have about a characteristic that's not in the fossils, it's not in the archaeology, it's completely from the gene, but it gives us a very large window of insight into what their adaptation was.
Red hair is thought to be an adaptation to lack of sunlight. Neanderthals lived in Ice Age conditions under leaden skies. It's hardly surprising they may have evolved the same adaptation. But the DNA findings suggest the Neanderthal version of the MC1R gene is subtly different from the modern human version. Because red hair appears to have developed separately in the two species, there's no evidence here of any interbreeding. But another gene's been discovered that's pointing to a different conclusion. A language gene has turned up in Neanderthal DNA, exactly the same version as in modern humans. Scientists are puzzled. It shouldn't be there. It may be the smoking gun they're looking for. Evidence that genes really could and did jump from one human species to another.